Hello everybody, my name is Omar and today we're going to explain the ovulatory cycle of fertilization. Fertilization is a precise multi-step biological process where sperm and an egg unite, leading to the formation of a zygote and initiating the journey of embryonic development. It's important to understand fertilization is fundamental for fields like reproductive endocrinology, fertility treatments, contraception, diagnosing early developmental issues. A common mistake is that some people confuse fertilization with conception. Fertilization is the union of gametes, while conception broadly refers to the formation of a new individual, which begins with fertilization but includes implantation. Implantation meaning that the zygote implants on the endometrium or uh, settles. I'm going to go over some key definition and concepts such as secondary oocyte, which, as we mentioned before, is a female gamete that is released during ovulation. It arrests in metaphase 2 of meiosis 2, typically fertilized in the ampulla of the fallopian tube. Now, what is an ampulla? An ampulla is the widest part of the fallopian tube. As you can see, we have the female uterus, there's an ovary and fundibulum, which are limb-like structures that pick up the secondary oocyte, and through the cilia that's found on the fallopian tube, there is propagation until it reaches the ampulla. Ampulla is the widest part, and there's the isthmus, which is the narrowest part, anatomically speaking. And the sperm would also move by the cilia till it reaches the ampulla where fertilization occurs. Now, this is an important key topic because it's asked a lot. After that, after fertilization occurs, implantation will occur on the endometrium. Zona pellucida is a thick, transparent extracellular glycoprotein, as we said, surrounding the oocyte. It contains a receptor called the ZP3 protein. These ZP3 proteins allow the binding of sperms. Corona radiata is a layer of granulosa cells that immediately surround the zona pellucida, providing nourishment and protection to the oocyte. So as we said, there's the zona pellucida, and then there is a corona radiata surrounding it. Seminal vesicles are glands found in males that contribute 60 to 70% of seminal fluid. Their secretions are rich in fructose, which is the primary energy for sperm motility, and prostaglandins, which are molecules that stimulate uterine contraction to aid sperm transport, and coagulase, which is an enzyme or called vesiculase, which helps seminal fluid coagulate in the vagina, preventing immediate drainage. Prostate gland contributes 30% or sometimes 40 of seminal fluid. Its secretion contains citrate, another nutrient for sperm, and fibrinolysin, which is an enzyme that later liquefies the coagulated semen. So it does the opposite action of coagulase and allows the sperm to become motile again. And a specific molecule called the prostate-specific antigen PSA. Sperm head. The anterior part of the sperm containing the haploid nucleus is 23 chromosome and is covered by the acrosome. Acrosome is a cap-like organelle, and the top of the sperm head is essentially a modified lysosome filled with hydrolytic enzymes such as hyaluronidase and acrosin, a protease. These enzymes are vital for penetrating the egg's protective layers. A midpiece is the section connecting the head and the tail, packed with numerous mitochondria that generate the ATP for flagellar movement. Flagella, or the tail, is a long whip-like structure responsible for sperm's motility and propulsion through the female reproductive tract. Fascitation, a series of physiological changes that the sperm undergoes in the female reproductive tract, uterus, or fallopian tubes that increase their motility and prepares them for acrosomal reaction. It involves the removal of cholesterol and glycoproteins from the sperm's head membrane. Acrosomal reaction, which is a process after capacitation, it's the exocytosis of the acrosomes enzymes triggered when a capacitated sperm binds to the ZP3 proteins of the zona pellucida. This allows the sperm to digest a path through the zone. Polyspermy is the fertilization of an egg by more than one sperm, which leads to an abnormal number of chromosomes and is typically lethal to the developing embryo. Now, physiological changes that occur to prevent polyspermy is called fast block or slow block. Fast block, which involves sodium, slow block involves calcium. It's a rapid temporary electrical change in the oocyte's membrane potential that occurs immediately upon the first sperm's fusion, depolarizing, which means making it more positive, depolarizing the membrane and preventing other sperm from binding. Slow block to polyspermy or cortical reaction. It's a slower but more permanent biochemical change initiated by calcium influx into the oocyte, leading to the release of enzymes from the cortical granules that modify and harden the zona pellucida, effectively blocking further sperm entry. Pronuclei, the haploid nucleus of the sperm, main pronucleus, and the haploid nucleus of the ovum, female pronucleus, before they fuse. Zygote is the diploid 2n46 chromosome cell formed by the fusion of the male and female pronuclei, representing the first cell of a new individual. Step-by-step explanation: sperm's journey and conditioning. After ejaculation, millions of sperm embark on a journey from the vagina through the cervix and uterus into the fallopian tubes. They face challenges like acidic vaginal environment and cervical mucus. As they travel through the uterus and fallopian tubes, sperms undergo capacitation, which occurs before acrosome reaction. This process involves the removal of cholesterol and glycoproteins from the sperm's head membrane, which makes the sperm more motile and prepares its acrosome for reaction when it encounters the egg. Penetration of the corona radiata. Capacitated sperm use their hypermotility and enzyme called hyaluronidase from the acrosome, which breaks down hyaluron to disperse the cell of the corona radiata. Binding to the zona pellucida and acrosomal reaction, a capacitated sperm binds specifically to receptors on the ZP3 protein of the zona pellucida. This binding triggers the acrosomal reaction, in which calciums, such as Ca2+, rush into the sperm, causing the outer acrosome membrane to fuse with the sperm's plasma membrane, releasing hydrolytic enzymes like acrosin and hyaluronidase. These enzymes digest a path through the zona pellucida, allowing the sperm to approach the oocyte membrane. Sperm oocyte membrane fusion. The first sperm that successfully digests a path through the zona pellucida will then bind to and fuse with the plasma membrane of the secondary oocyte. The entire sperm enters the oocyte cytoplasm. As we said, fast block to polyspermy, in which a rapid temporary electrical block as the first sperm membrane fuses with the oocyte, sodium ions Na+ rush into the oocyte, causing its membrane potential to become positive. This rapid change immediately repels other sperm from binding to the oocyte. Slow block is or cortical reaction, slower permanent biochemical block, in which it involves Ca2+ released from the oocyte smooth endoplasmic reticulum. These Ca2+ ions trigger exocytosis, which means like expulsion of cortical granules or lysosomes, which are type of breakdown molecules located just beneath the oocyte's membrane. These granules release enzymes that degrade the ZP3 proteins. This breakdown will lead to a hardening of the zona pellucida. Breaking down the ZP3 protein will make the zona pellucida a more hardened structure, sealing the egg from further sperm entry. The influx of calcium also signals the secondary oocyte to complete its second meiotic division, which was arrested at metaphase 2, forming a mature definitive ovum and a second polar body. The nucleus of the sperm decondenses to form male pronucleus, and the nucleus of the newly formed ovum forms the female pronucleus. These two join together, restoring the diploid chromosome number and forming the zygote, the first cell of a new individual. Worked example: blocking polyspermy. A closer look. A researcher observes an in vitro fertilization experiment where multiple sperm are able to penetrate the zona pellucida and fuse with the oocyte membrane. What two critical mechanisms failed, and how do they normally prevent this? It means that why did polyspermy happen? It's because either both failed or one of them failed. But in this case, they're asking for two. It means that the fast block and the slow block have failed. It's which fast block, as we repeat, normally when the first sperm fuses, a rapid influx of sodium, Na+, changes the oocyte membrane potential from negative to positive. This electrical repulsion should immediately deter other sperm from binding or fusing. The cortical reaction or slow block, its failure means that the cortical reaction, a slower but permanent block, also failed. Normally, the influx of calcium's ions such as Ca2+ after sperm entry triggers the release of enzymes from cortical granules. Granules are basically like small package structures that have inside of them enzymes. These enzymes modify and harden the zona pellucida and degrade ZP3 proteins. Thus, when we're hardening the zona pellucida, we're basically removing ZP3, which allows binding of other sperms, making it impenetratable to any subsequent sperm. Final answer: both rapid electrical depolarization fast block and slow block have failed to prevent polyspermy in this scenario. Now, expert key takeaways and especially the significance of this topic is that fertilization is the precise biological event that marks the beginning of human life and genetic inheritance. That's why it's important. Where we might use this fertilization, it's critical for in vitro fertilization, which means the entire process of IVF relies on controlling and facilitating fertilization outside the body. In vitro means outside, in vivo it means inside. Contraception development. Many contraceptive methods aim to prevent fertilization by preventing sperm-egg encounter or inhibiting sperm capacitation. Diagnosis of infertility, identifying issues at any step of fertilization process such as sperm motility, egg quality, zone of pellucid abnormality is key to diagnosing and treating infertility. Understanding genetic disorders, errors during or immediately after fertilization can lead to chromosomal abnormalities such as polyploidy. What you would expect on the exam, expect questions of specific roles of sperm parts, acrosome, mitochondria, the steps of fertilization such as capacitation, acrosome reaction, and dual mechanisms for preventing polyspermy. And I can't emphasize this enough that this dual mechanism is really important and it's asked a lot on multiple exams. Thank you.